Hi there. Sorry for the brief interruption, just to let you know that the audio and video quality of that online meeting was not the best we ever had. So for once, you will not see the speaker in the small window where I'm right now. And if you find any presentation hard to follow, just reach out and we'll make sure your questions get to the right person. One more little thing. I was a bit late hitting the record button, so I missed the first 2 seconds of the first talk from Stefan about mass spec of RIT. So once more, if you have questions about that specific topic, just reach out and we'll make sure he goes to questions. Thank you very much.
We're using mass spectrometry to look at metabolites from different microbes, basically, trying to see if there's any antibiotics as part of that. We have Eli Borgo's lab, looking also at metabolomics, looking at targeted metabolomics, or I think lipids. And then we have Dr. Thomas' lab, working on proteomics with bacteriophages. And continuing on the proteomics theme, Dr. Kree's lab and my own lab as well. And then we have in the School of Chemistry, Dr. Schnitterner's group, working on peptides and fluorescent dyes. So basically using mass spectrometry to do chemical analyses of whatever they are using for their experiments. And then we also have a chemical analysis laboratory, kind of a core facility. Dr. Olson is leading that one. There's a bunch of mass specs there. I haven't had a chance yet to get a tour there. Maybe that's somewhere in the future. Yeah, we just need to convince. And then, and then in the Department of Biomedical Engineering, we have the Kozak lab. Kyle might know them because they've measured samples at UFR as well. They're working on extracellular vesicle proteomics. And then on the computer science side, we also have the Casey lab. Here, for predicting post-translation modifications and using mass spectrometry datasets, proteomics datasets as a foundation for training those models. So, in my opinion, a nice, diverse field of people working with mass spectrometry and mass spectrometry data. But importantly, also a lot of different students from various different majors working with mass spectrometry data. And so I wanted to use today kind of to provide a platform for those students, at least for some of those students, because I think it's a nice opportunity to have this network, to have this opportunity to present their work, maybe ask questions about mass spectrometry, maybe learn from all the experts in the rooms, get advice, troubleshoot, etc., and network, find community for later on in this stage as well. And of course, the other way around as well, and employers and so on, if they're looking for students for internships, for work later on and so on, might find some good ones. Hello, everybody. I am Giacomeli. I'm a fourth-year biology student here at RIT, working under Dr. Schulze. I'm here to present about sample preparation methods for comprehensive proteomics in bacteria and archaea. Before we do any methods or anything, we need to understand the samples that we're analyzing. The two organisms I worked with were Pseudomonas aeruginosa and Methiococcus maripalidis. Pseudomonas, gram-negative bacteria, we use cell fractionation. Methiococcus, archaea, we use whole cell extraction. For cell fractionation, there's a couple things to think about downstream, since you're taking the individual components of bacterium, namely the extracellular cytosol and the membrane. Each fraction showing is going to be easier to map the peptides. versus for the archaea, the whole cell extraction, you're putting the whole proteome into a single sample, so it's going to be increased computational. Why are we using different licenses? It's based on the specific purpose of the analysis that we're doing. For Pseudomonas aeruginosa, we are trying to find glycosylation patterns in wild type and knockout strains. So subcellular fractionation works very well for this, as it creates a reduced complexity, which makes it much easier to find specific glycoproteins and an analysis. For Methanococcus, we are trying to find metal ion chaperones grown at different concentrations. So whole cell lysis for this works very well. In the very end, it maximizes quantification accuracy because it reduces the variability between samples. I have a figure from Hensenberg et al., a paper that I will continue to work on, just showing the glycoprotein on Pseudomonas PA01 specifically. The important aspects of sample preparation are as follows: data quality and reproducibility. Every step on sample preparation has its effects downstream for finding analysis. So that could be contamination, that could be errors in calculation. All of these things have direct consequences. And just a nice figure I found for a comparison between clean sample and plastic contamination PPG for on spectra. Reproducibility, cornerstone of science. If you can't do it again, is it an outlier or not? Here's a basic sample preparation workflow, starting with sample collection, lysis, protein quantification, STS stage, S-trap, and then O1 mass spec analysis. Starting with Methanococcus, which we did a whole cell extraction on. It has 5 pretty simple steps, starting with protease and phosphatase inhibitors. This prevents any protein degradation or any further reaction from occurring. From this, we lyse the cells. We specifically use the sonicator to use sound waves to break open the cells. From there, we add SDS to 2%, which will solubilize the proteins. We incubate that for 55 degrees, which we'll put into solution. From there, we can centrifuge it. The purpose of this is to concentrate it as well as get rid of all of the contaminants that can possibly be in there. And from there, you have your sample. Following this for Pseudomonas, the cell fractionation, it's very similar, however, with a couple extra steps. To begin, you take your sample and centrifuge it. You'll get your supernatant and your retinate. Your supernatant is going to be your extracellular fraction, so you need to concentrate this to get rid of all of the broth elements. With the cell pellet, you then lyse it and resuspend it, put in ultracentrifugation, and it separates into the pellet and the supernatant. The supernatant will be the cytosol, and the pellet will be the membrane. From here, you do the same as before, and you solubilize them, incubate them, and you filter them. Following this is our protein quantification, specifically the BCA assay. This ensures consistency across all the samples, as well as picks out any possible errors that might occur throughout. Right here, I have the membrane cytosol supernatant out of specifically the Pseudomonas strains that I did: PA01, PA14, CW1, and CW5. CW1 and 5 are knockout strains, specifically via transposon, and PA01 and PA14 are the wild type strains. The supernatant, as you can see, has a very low protein amount. That is understandable, given that is the extracellular, while the cytosol, where all the metabolic processes go down, have the highest concentration, and the membrane being low. Following protein quantification is quality control, which means SES stage to visually analyze the lysis, extraction, and integrity of the proteins. These are the same as before with the BCA, with the supernatant size membrane, and consistent. The supernatant is very low bands, as expected, since there's very low protein. Membrane following close, and cytosol with very deep bands, although some inconsistency. Following this, S-trap digestion, which, as we all know, is for cleanup and breaking down the proteins into peptides. I really like this image right here. It illustrates how different digest can give different results based on in combination or if they're used by themselves. I specifically use trypsin or metrag for creating our enzymes. The specific machines we used for our mass spec was the TIMS TOF Pro for the Methococcus and Thermo Fisher Orbitrap Eclipse for the Pseudomonas. This is our very simplified bioinformatic analysis yet where you take raw data, you take that, convert it to MFD, put it through database searches using Xtandem and MS Fragment, as well as another. Validation by putting it against fake results. And then you filter where you combine and exclude specific data. If you're looking for specific modification, this is the spot where you would show it against others. For our preliminary results, my Pseudomonas aeruginosa, there was about 2,500 proteins out of 5,500 that are predicted, equaling about 42% of the whole proteome, while Methanococcus had 1,400 out of 1,700 predicted, representing about 82%. This makes a decent amount of sense. Methanococcus has a much less complex proteome versus Pseudomonas, which has a very complex proteome, and a lot of their expressed proteins are very specific to the time-like growth phase and as well as environmental conditions. Following this, the specific, like my modification I was looking for was N-dimethyl L-threonine. This was specified by Hensenberg et al., where they took an old paper Germa et al., who hypothesized a glycoprotein modification on B-type flagellants, and that's their proposed structure with a L-rhamnose phosphate group with a dimethyl threonine. Then my results, we got 5 hit total for this modification, all being within the wild types and none detected within knockouts. One thing to note is that these were not on any of the beta-type flagellants. And this makes sense though, because they used a K proteinase versus trypsin. So it wasn't expected to genuinely find it. I would like to acknowledge Dr. Stefan Schulze in the Schulze lab at RIT, Kylie Allen in the proteomics core facility at Virginia Tech, and the proteomics core facility at University of Ahmed.
Hello, and it's Kopelev. I'm the researcher in Dr. Schulze's lab. I'm also the president of Bioinformatics Club. I don't mean anything, but I try to get into the field more. Dr. Schulze, as the present today, thank you for the opportunity. I'll be talking about a project that is it's mostly in UPenn, I believe. But I'm doing a small project that goes deep to the project, so I'll just be covering that. So quantitative analysis of gene influence on protein mobilization and alloperic proteomics. An overcap of archaea. Archaea's relationship to eukaryotes. So people used to believe that archaea was a distant relative of eukaryotes, but then as we can see right here, newer hypothesis says that eukaryotes actually stem from archaea. So there's more research being done on it because we want to know more about eukaryotes. We want to know more about archaea. We're specifically studying alloperic volcani. Why are we studying? It's easy to culture. Its ability to grow on defined media and amenability to genetic and biochemical. Also, there's a huge community surrounding it. So the more people study it, and the more data there is about it, the more we can cross-reference on the market. Right? Lipidation to lipoproteins. Lipoproteins are lipids.
combined with proteins, attached lipid molecules attached to protein. It's observed across all domains. An important thing to note is that lipoproteins affect hydrophobicity, enzymatic activities, and subcellular localization. So, as we can see right here, lipidation, it affects how an organism communicates, adhesion properties, morphology, chemotaxis, motility, and nutrient uptake. The reason that we want to look at lipidation is that when we look at lipidation in eukaryotes, studies have shown that it has connection to a lot of cellular processes, such as cell death, protein trafficking, and cancer development. So if we want to know more about eukaryotes, and archaea is connected to eukaryotes, that means looking at lipidation in archaea helps us understand lipidation. So lipoproteins in archaea. Lipidation exploration in archaea, identifying and measuring lipidated proteins and the genes that control it. We want to compare this mechanism. Once we understand this mechanism, we want to compare it to eukaryote lipidation. Protein lipidation in archaea remains limited compared to that in bacteria and eukaryotes. Okay, so what do we know about lipidation in archaea? The master thesis project this is based on is after it was found that the specific lipidation mechanism in archaea, specifically with ALI-A and ALI-B genes. So the current study, this is the study that is happening at UN. It's discovery of the first archaeal lipoprotein biogenesis. This study confirms the distinct substrate specificities of ALI-A and ALI-B, analyzed the lipidation status of predicted lipoproteins in ALI-A, ALI-B, and so knocking out ALI-A, knocking out ALI-B, and whatnot. It overall establishes ALI-A as the primary enzyme responsible for archaeal lipoprotein lipidation. The wet lab side includes growth, cell lysis, Triton X extraction, and mass spectrometry preparation. This is more of what I was my contribution to the project. It was peptide protein identification using OSCAR framework, and it used those enzymes, the MS Brager, Tandem, and MSGF. And then for identification and for quantification, we used the OSCAR framework using Flash LFQ. For the quantification part of this, there was an MS Stats pipeline used for this. The MS Stats is a R-based package, and there's additional steps, but for this specific contribution, we went through data pre-processing, exploratory data analysis, model-based analysis. We didn't go into small scale or large scale. That we were just trying to understand the actual abundances, the specific proteins, just to sample. A quick note about this MS Stats workflow includes the small scale and large scale, which it's standardized workflow. So for other data sets, we can compare and add to the Archaeal project. That's different. So the first thing in this workflow is the data pre-processing. So this just involves taking that quantification CSV files from the Flash LFQ output and just formatting it, because MS Stats needs specific formats. So that's basically just like deleting columns, adding columns, just framing. adding 4. Exploratory data, here we can see normalization, then we can see the peptides, and then we can see the peptides summarized. Here we can actually see the significant proteins in the moving outplot. This results EMAP to represent this aspect, but this has to do with the model-based analysis. So this is what I've been working on. This has to do with looking at the wild type and looking at the proteins compared to predicted proteins and the numbers per feature. So right here we can see the wild type and the TX, which is tretinoin X protein. So this is looking at membrane bound protein. Sorry, because this helps extract the membranes, the proteins within the membrane. And this is just so this is this is without. One of the important things when doing a study like this is establishing a baseline. So these numbers are the foundation for this type of study. So we have confirmed here that knocking out through the wild type, both knockouts, ALI-A and ALI-B separately, it confirms the genes ALI-A and ALI-B, their role in evolution. References, acknowledgements, Dr. Schulze lab, and the UPenn group. Yeah. Thank you for listening.
Hi, my name is Nikhi Boggavarapu. I'm a 4th-year double majoring in biotechnology and bioinformatics here at RIT. And I've been working on this project for about a year and a half now, part of the Schulze lab, doing a collaboration project with the Wasred lab. And our project is trying to do a comparative proteomic analysis of multiple Neisseria gonorrhoeae strains with the overall goal trying to figure out what the antibiotic resistance mechanism that's occurring in Neisseria. So a little bit of a background about Neisseria. Gonorrhoeae, it's a well-known STD that is quickly developing resistance to drugs like gentamicin and vancomycin, which have been commonly used to treat gonorrhea, but slowly with the increasing resistance, the drugs are not proving to be as effective. And almost 106 million new cases of gonorrhea are diagnosed annually. So this is a significant problem in the population. So this gram-negative bacteria has multiple external proteins of pili, and this pili is important to gonorrhea because it allows it to attach to the sperm cells, which allows transmission between hosts. While alternative therapies are working on being developed, like bacteriophage therapy, a lot of work still needs to go into that. And while that's working, the resistance is just getting worse in the strain. So we want to take a proteomics approach to try and answer that question. So I chose to take 2 approaches for my project by utilizing a wet lab and a dry lab approach. So looking at the wet lab, what we did was we exposed gonorrhea to penicillin over a time point 0, 2, 4, and 8, and we collected samples at each time point to then be able to do cell lysis and collect the proteins from the cells, which were then sent to the University of Rochester to collect mass spectrometry data. And then we look at the dry lab where I perform a bioinformatics approach that involved optimizing the data, making sure it was set to be able to do analysis, and then quantifying it so we can make more meaningful conclusions with the data. So our isolate of interest is FA6140. FA6140, the way the resistance works in this, I'm sorry about the formatting. It was looking good on my laptop, so please bear with me. Yes. So the way resistance works in this strain is it's growing pretty well at first. However, after penicillin is added, the number of living cells actually decreases. However, it doesn't hit 0. That's because some cells have developed this resistance mechanism called factor X. And all the new cells growing after all have this factor X mechanism. The reason why we call it factor X is because we don't actually know what's causing this resistance mechanism. Resistance in FA6140 is unknown at the genetic level, which is uncommon when studying antibiotic resistance. So we want to see if proteomics can answer the question that we still have. So this is the growth curve I was mentioning earlier. FA6140 is in blue, and as you can tell, it increases. It's kind of slowly decreases and does ultimately die off at time point 8. But at that point, that's when it's really pushing its limits with in regards to nutrients and what it survives in. And then we also work with 3 other strains. AZOI, which is our highly resistant strain, that's referring to the green one. As you can tell, it's very high up at time point 4, and it doesn't even die off by time point 8. So it's a really resistant strain. And then we have SP111 and SP114, which are both which are sister strains that are more susceptible to penicillin, so they die off pretty quickly. So, oh god, sorry guys. Yeah, so as I mentioned, I work with 4 strains. AZOI is a positive control, and it's actually very similar to FA640, which is why it's such an important strain, because subtle differences between FA6 and AZOI will point to proteins that are of interest for antibiotic resistance. And then SP111 and SP114 are also related to FA6, albeit not as closely, and are acting as R negative controls. So, just a reminder, we took samples at 0, 2, 4, and 8, and we added penicillin after time point 0. So we'll see the effect of penicillin when we analyze data for time point 2, 4, and 8. After collecting the samples, we did viability assays to basically confirm that cells were growing as we expected it. And then we prepped the samples and sent them out. So this is the overall schematic we were going for when designing the project. Essentially, we're going to be doing, we did an analysis between all the strains and all the time points. And the goal is that we're able to find some pattern between all of these comparisons that answers our question, again, which proteins, or which group of proteins, which type are contributing to resistance. So then we've got sample prep for mass spec, pretty standard. And we sent those samples over to the University of Rochester using their Orbitrap Astro mass spectrometer. And we got that data back, and I've been doing analysis with that since. And this is measured with label-free quantification using a data-dependent acquisition approach. So looking at the bioinformatics methods, first we look at protein identification. So we use Erscall, which is a Python framework that creates a workflow between search engines to statistical processing, and search engines were, like I've mentioned in the earlier presentations, MS-Fragger, MSVF+, and Xtandem. And these were used for identifying the proteins and then just doing basic analysis. But next step was quantification, where we use FlashLQ. It's a label-free quantification tool, and it calculates intensity values. However, intensity values aren't enough, and we need to do more data analysis, where we use MSStats. MSStats calculates the longitudinal volume between the proteins across various samples. So I did mention how I was comparing all those various conditions before. The image on the right shows all the different comparisons I did. I believe there was like, I don't know, close to 30 of them. It was a lot, but it was important because all those different condition comparisons answered different small parts of the question. Like, it all adds to that story, and it's incredibly important, even though it's a lot of data to look at. But it was great because I've been using R primarily to do all my data analysis, and it's such a useful tool. I love it. So looking at the analysis, using the fold changes, we're going to be able to compare how the protein changes from its standard. So we created a lot of plots to be able to really visualize this. So we use Fsplots. Our primary focus, as I mentioned earlier, FA6140 and AZOI are very similar. So we really want to observe those changes right in the stream. So this plot is setting AZOI 10.0 versus FA6140 10.0 as our baseline, as our control. And then it plots all the other proteins in the other time points. So AZOI 10.2 versus FA6 10.2. and it allows us to visualize how different the abundance are from its standard. So this allowed us to create a list of proteins that we might want to take a further look at. Points that are lower than the green line are ones that have a lower abundance than our standard, and then the points that are above the green line have higher abundance than our standard. However, this is a bit of a static graph, and we couldn't really understand how significant the changes were, so we decided to utilize heat maps. Here's an example of the heat map. This is using the same conditions as I used on my X-plot. But what we look for in a heat map is highlighted by the state box. There's one consistent color across all conditions and multiple proteins. So the x-axis are the proteins, the y-axis are my conditions. And that's what we look for when we do the heat map. So zooming in on that, we notice that it's all like multiple shades of blue. Blue is referring to a lower abundance, or lower potency, so lower abundance. And seeing all those like closely isolated proteins makes us consider like these proteins could really be involved with the resistance factor. One of the proteins actually involved is, not involved. One of the proteins that shows up here is a PEL-E protein, which is a pili protein, again, associated with pathogenic cells. So, we have to do further analysis to see what that means in our, in the story we're trying to explain. But it's nice because we're able to kind of really understand how significant these changes are towards answering our question. And then we've been using BS Plus to do further analysis. And BS Plus is a tool that allows us to cluster the proteins into groups. It has an algorithm that tries to detect patterns and how the abundances changes in the protein and match it with other proteins. And then it uses statistical analysis to determine if that pattern is statistically significant. So this is pretty useful because the idea is we can hopefully get a group of proteins that are acting all similarly when they contribute to this extent. And we're able to map out all the conditions. So this is basically separating all the proteins we got data on into 8 different groups and just showing how these proteins interact. And as you can tell, they all have a similar pattern. So our next steps require like looking at the heat maps for each of these cluster plots and seeing if it really reflects true significance that's also associated with changes in abundance. So those are the next plans for the project. And our goal is by looking at those heat maps, we'll be able to find, again, those less of proteins, and hopefully we can do further analysis in the future. But yeah. So special thanks to Dr. Schulze for taking me on and helping me with intensive parts of this experiment. And thank you to Dr. Wadsworth for assisting with the turnout in Neisseria gonorrhoeae. I'm going to be talking to you about one of my projects in the lab, which is the incorporation of methionine sulfoxide into nascent proteins. So I'll just jump right in. So I think everybody has like a biology background. So you probably know that methionine is one amino acids used to, methionine is one of the amino acids that's used to make proteins. And methionines are special in that reactive oxygen species that are generated from byproducts of cellular metabolism, which are just like oxygen-containing molecules that are highly reactive. If those reactive oxygen species react with methionine, they can convert methionine into methionine sulfoxide. And this oxidation can happen and create 2 different diastereomers, which is methionine R sulfoxide and S sulfoxide. But basically, this changes methionine from a non-polar amino acid to now a polar amino acid. And this has different consequences for the proteins that the methionines are found in. So as I mentioned, there are some different consequences of methionine oxidation. And here's just a schematic of a few different things that people have discovered methionine oxidation plays a role in. So if methionine in a protein becomes oxidized, in one case, people have shown that this can lead to aggregation of proteins as those now they used to be hydrophilic molecules. Now they're going to be aggregated to hide those potentially other hydrophobic amino acids that have now been exposed. Methionine oxidation may also be a signaling event. So if a methionine protein becomes oxidized, that can allow it to turn on in a sense and not performance function. And then methionine oxidation has also been hypothesized to act as a reactive oxygen species scavenger. So methionines and proteins can react with those reactive oxygen species and protect other residues around those methionine from becoming oxidized. And as I mentioned before, methionine oxidation, or as I just mentioned, they can have either deleterious consequences or it can be a functional beneficial consequence in a sense. But interestingly, in cells, there are also these enzymes called methionine sulfoxide reductases. And these are conserved from bacteria to eukaryotes. In bacteria, there are 5 different versions of these methionine sulfoxide reductases, or MSRs. Here I've shown the structure of them below. And bringing back to stereochemistry, these different enzymes work on different stereoisomers of methionine sulfoxide. And some of them work on either peptidyl or free methionine sulfoxide. So either methionines in proteins or just free methionines splitting the cell. And because of this whole system of the reduction of methionines and also the consequences that methionine oxidation can have, our lab and many other labs have been interested in studying methionine oxidation. And another just like biological significance is that if a methionine or protein became oxidized, that could lead to misfolding. As I mentioned before, where now the hydrophobic residue is hydrophilic and it wants to fold in a different way. And this can cause aggregation, and that has been linked to different neurodegenerative diseases like Alzheimer's, and may also just be linked to pathological aging. So just like establishing kind of why we're interested in methionine oxidation, I'm going to explain. So a lot of this work is focused on methionine oxidation in proteins, but I'm going to be explaining a different aspect of this oxidation. So our big question is, can you take a free methionine sulfoxide and can that be added onto a tRNA through amino acylation and then make it to the ribosome and actually get incorporated into a protein through translation? Whereas like a lot of the other projects in the past have focused on actually looking at oxidation as a post-translational modification. So I've been working on this project, or when I joined like 3 years ago, I joined the project working on the in vivo side of this experiment, but I'm going to talk about first like the in vitro experiments that kind of were the basis for my experiments. So first, just to verify that you could even incorporate methionine sulfoxide into a protein, we had this in vitro translation and transcription system. So we had the plasmid or template for a model protein, and then all the different factors necessary for transcription and translation, such as polymerases and other transcription factors. Then we also added the tRNAs and amino acids. And then here, what we did was we either added methionine or methionine sulfoxide. And we want to see if our model protein could be produced. And in the assays that I'll show you later on, we're using GFP as a model protein. So if protein was being synthesized in this assay, you'll see fluorescence, and that's what we can observe. So here's kind of what I was talking about before. This assay, for this experiment, we had our in vitro translation system. We either added methionine, which is designated by the blue line, or methionine sulfoxide, which is the orange line. And then also we had a control just to see if there was any background fluorescence that we pick up over time. And from here, you can see that there is some, with methionine, you obviously get protein being produced, you get a good signal of fluorescence. And then with methionine sulfoxide, there is a slight increase in protein production, but it doesn't look like it's very efficient. So this kind of shows us that in normal standard conditions, matching the concentration that enzymes are normally in the cell, you can't really efficiently incorporate methionine sulfoxide into a protein. And now, I don't remember if you remember what was on the schematic beforehand. So methionine, or METRS, is what's designated above. That's methionine tRNA synthetase. So this is the enzyme that will add methionine onto a tRNA so that it can be incorporated into a protein for translation. So that's basically the first barrier. If methionine sulfoxide couldn't get into a protein, this would be the first step that could be possibly the barrier for its entry. So when we overexpressed or kind of over-supplemented methionine or METRS in this system, you can see that the green line, which is methionine, it's about the same amount of protein product being produced in this assay. But now with more METRS, we get a lot more protein produced in the methionine sulfoxide treatment. And this is showing us that while methionine sulfoxide might not be readily incorporated into a protein, there are ways that you can forcibly introduce methionine sulfoxide into a protein. translation. So once we know that this is at least possible in vitro, my project was really looking at if you can take methionine sulfoxide and add it to like cell media and have that incorporated into a protein in a cell. But there are a few issues, just like technical issues, that would come up in this case. The first thing is that if you took the proteins from a cell and you do the normal mass spec processing or sample processing, and you run your peptides through the mass spec, there's a lot of artifactual oxidation that could be introduced, just through like the electrophilic ionization process, or even like maybe spitting down the feedback your samples. So this is just like an illustration I have. So like this might be the normal distribution of like if the orange was like how much methionine sulfoxide containing peptides, and then blue were methionine containing peptides. This might be what is actually in a cell, but then if you got your data back, this is what it could look like, where there's a lot more artifactual oxidation, and you just see a lot less of your methionine. And that would kind of skew the results. So you wouldn't really know, is the methionine sulfoxide that in the cell being incorporated, or is it as efficient as this result is showing? That's one issue, just technical issue. And there's two other issues that come with working with living organisms. So bacteria, I'm going to be working with E. coli for the rest of this project. And E. coli have a methionine biosynthesis gene, so they can produce their own methionine. So even if I just gave them methionine sulfoxide in their media, so that's the only thing they should be able to grow on, they can technically use the nutrients in the media to make their own methionine. And then again, like I mentioned before, there are these MSR enzymes that can reduce methionine sulfoxide, and that could also impede the results. So maybe if the methionine sulfoxide was incorporated, it was then subsequently reduced. And then in the proteomic data, it doesn't look like it was ever incorporated. So to get over these two problems, we came up with two solutions. So the first thing is that we have this E. coli strain that our collaborator generated for us, actually. So we took out one of the methionine biosynthesis pathway genes, which is MET-P or their orange. And we also removed the genes for all these 5 MSR enzymes. So now this strain should not be able to reduce meto, and also it will need methionine in the media in order to survive. So in the subsequent experiments I'll show, the experiment is one where I have a 1-to-1 ratio of methionine and methionine sulfoxide. But then since you still wouldn't be able to tell, like if I just had a normal methionine sulfoxide and then normal methionine, again, you wouldn't really be able to distinguish if something happened, like if somehow stochastically it was reduced and you just see regular methionine in the cell. So in order to make sure that the methionine sulfoxide that we added to the cell was actually incorporated, what we did is we took this isotopically labeled heavy methionine. So all the carbonates in the backbone are C13. And then I oxidized it with an O18 hydrogen peroxide. So this would oxidize that sulfur, making methionine sulfoxide that is now double labeled. So the backbone is labeled, and then also the oxygen is labeled. So any peptides that have this methionine sulfoxide incorporated into it, we can be sure that that was methionine sulfoxide that was actually incorporated, and not just a methionine that became oxidized or that it wasn't from any other source. So in the data I'm going to show you, this is kind of like the 4 different types of methionine that you could see. So I am adding like a 1-to-1 ratio of like light methionine and heavy methionine. So you will get some like normal C12 methionine in the mixture. But if what we hope to happen is actually happening, you would see this C13 and O18 methionine sulfoxide, which is designated red. But I guess there could also technically be, even though we remove those MSR enzymes, there is a possibility that you can reduce that methionine sulfoxide. So you could just get like the heavy backbone labeled methionine, and that could be what you see in your protein. Or there's another case, since I'm not going to be accounting for any artifactual oxidation in this method, you could have the case where the methionine becomes reduced and then subsequently oxidized, either in the cell or through like the sample processing. So this is just a wild type strain. So this strain has MSRs, as drawn by the plus at the bottom. So if I have a normal amount of that meta-RS enzyme, which amino isolates methionine onto tRNA, you get a pretty big signal of normal light methionine. And then you also see a lot of that yellow, which is the reduced methionine sulfoxide, which is what we would expect if these cells have MSRs. But if you can see once we overexpress NRS, you still don't really get an increase in that red. Like the red would be at the top, but you don't really see that at all. So that kind of shows that there's no incorporation of methionine sulfoxide. And in this case, it's all being reduced, and then it could have gotten oxidized afterwards, as shown by the orange. But now for our better control, where we don't have those MSR enzymes, and there should be no way that the meadow is becoming reduced. This is what we see. So all of the methionine in the proteins is actually the normal methionine that we added to the media, and none of those methionine sulfoxide are becoming reduced or incorporated. And that kind of shows that there's something else in vivo that even if you overexpress NRS, there's something else that's blocking those methionine sulfoxide from getting into protein. So this is kind of just like a schematic or model where we've looked at the other proteins downstream from amino isolation, but I've only done that first step in vivo so far. But basically, if you overexpress NRS, that's one of the biggest barriers, or you get over one of the biggest barriers to methionine sulfoxide incorporation, at least in vitro. But in vivo, it seems like even if you overexpress NRS, there's something either blocking it from even being amino isolated or downstream from being incorporated into protein. And we're still doing some experiments to figure out exactly what step is the barrier. But this is kind of our working model right now. And I have this other like side project that kind of came out of all the data that we collected from these experiments. So we just want to see if when we're adding MET-O into the media for these cells, we just want to understand what that's like kind of doing to the cells and how that changes protein expression patterns in E. coli. So this is grown in a different condition from those last experiments. And what we noticed was that actually in anaerobic conditions, or like slightly more anaerobic condition, we grew this MSR mutant E. coli strain in different concentrations of MET or methionine sulfoxide. And you can see that this is a log 10 scale, so this is actually 10 millimolar. But at its concentration of methionine sulfoxide, you can actually get the cells to grow on just methionine sulfoxide. So even though these cells should not have any MSR enzymes and they shouldn't be able to make their own methionine, if you add enough methionine sulfoxide, they can grow on just this. So we want to understand kind of a little bit more what's happening here. Like, is this becoming directly incorporated, or is this methionine sulfoxide being reduced? So we use the same method as I used before. And this is just looking at that treatment which has the highest level of methionine sulfoxide added. And now what we see actually is that there's a lot more of that methionine sulfoxide becoming reduced and some background oxidation possibly. But again, there's no direct incorporation of methionine sulfoxide, which was very interesting to us because the strain shouldn't be able to reduce those methionine sulfoxides. So what we want to do, again, using our proteomic data set, is look at any expression level differences to see if there's any patterns there. And what we actually saw was that this whole class of enzymes in the DMSO reductase family are being upregulated in this MSR mutant strain compared to a wild type strain. And I'll go into a little bit more what those DMSO reductases are. So normally normally DMSO, which is dimethyl sulfoxide, becomes reduced by those DMSO reductases. And those different genes over there that are being highlighted by the stars are different subunits or gene duplications of different subunits of the DMSO reductase. Which you can see that sulfoxide group on DMSO looks very similar to methionine sulfoxide. What actually shown in the past that DMSO reductases can work on methionine sulfoxide and reduce that back to methionine. So our model for this system right now in anaerobic conditions, which DMSO reductase are usually upregulated in anaerobic conditions to begin with because it can act as a terminal electron acceptor for anaerobic respiration. So what we're thinking though for these MSR mutants, they have some other way of sensing that there's a need for like this DMSO reductase to be overexpressed, and they're overexpressing those DMSO reductases to kind of compensate for the introduction of methionine oxidation that we have given to the cells. And that's kind of just like an overview of my project. I want to thank my lab and the mass spec research and our collaborators.
So in principle, as you all know, you generate proteomics datasets to answer biological questions, right? And normally each data set kind of tries to answer a specific question. So you have all of those individual data sets generated for those individual biological questions. And a lot of them end up on the product guide, on public databases, storing proteomics data sets. What we are trying to do is to bring all of that together, to combine those data sets, reanalyze them in a unified way, and then combine the data that we get from that, the results that we get from, or to be basically link the different biological questions that those were generated for, to get more biological insights. And for that, on the Archaeal side, we have initiated the Archaeal Proteome Project, where we combine the data sets, also combine them with expert knowledge. So we contact the groups that have generated those data sets to see, how they did it and what insights they got so far. As a first part here, importantly, all of that helps, of course, to increase the overall proteome coverage. So the more data sets we have, the more proteins we can identify, the better we understand really which proteins are actually expressed in the cell. It also helps us with sample preparation. We can compare the different data sets and see how they were generated, which sample preparation they used to get the highest proteome coverage. Also, for example, at this step here, that was after we had done the first version of the Archaeal Proteome Project, we generated a new data set based on the improved sample preparation that we learned from this, and could increase our protein coverage quite a bit there. Secondly, proteome coverage in the sense of identifying proteins across the proteome is of course important, but even more importantly for projects that I'll go into in a second is to also increase the sequence coverage, meaning for each protein, how much of the sequence do we actually identify? And that had an even higher bump, at least relatively speaking, right? So we go from somewhere in the 20% to more than 40% in our sequence coverage. Really doubling it, more than doubling it. And that is important, especially for the analysis of post-selection modifications. In our lab specifically, we are interested in protein vaccination. And of course, we can only see protein vaccination if we identify the peptides that are modified. So for that, we need a high sequence coverage. And we are interested in protein vaccination because it's involved in a lot of important biological processes. A lot of you probably know it from eukaryotes, where it's involved in everything that is important. But it also occurs in prokaryotes, and it's important for motility, bioconformation, mating, cell shape, various different things. So with the Archaeal Proteome Project, with that increased sequence coverage, we could increase our number of identified glycoproteins roughly by a factor 6 or 8. We could increase our number of identified glycopeptides roughly by an order of magnitude. So really bringing together all of those different data sets from those different conditions with the increased sequence coverage and so on really helps a lot to see what is happening in the cells, see those glycoproteins and glycopeptides. So that was really heartening to us to see, and we wanted to see if we can expand that to other archaea, other prokaryotes, and we collaborated with a group in India that had generated the database, the ProBlackBot on Archaeal, and we were looking for any archaea for which the constellation has been described. And so all of those were in the database. We then checked the product guide if we see any data sets, any proteomics data sets for those archaea. Got a couple of them, and then used our bioinformatic workflows within the Archaeal Proteome Project to see if we can identify the glycoproteins. Again, sequence coverage is important. For those species where we have a high sequence coverage overall, we do check the glycoproteins, the glycopeptides. The better the sequence coverage, the more we identify, basically. And that's the gist of this one. And then also, for most species, we identify more than what was known before. So again, bringing together all of those different data sets helps a lot to learn more about the species. And this works for various different glycans. You might know for eukaryotes that all of the glycans for the different eukaryotes look very similar. because evolutionarily they are more related than the really diverse range of archaea that we have. So all of those crazy-looking glycans we're able to analyze with our pipeline. So just from a bioinformatics perspective, there's a lot of tools that are developed for glycoproteomics in eukaryotes. They all assume a common glycan core. None of them is useful for us. So we have to work with tools that are a bit more flexible in that sense. And it works. And then we also wanted to look at, well, if we can analyze all of those where the glycan composition is known, maybe can we also expand that to archaea where the glycan composition is not known. And that's what a couple of students were working on in the lab. Great Brian Theotismus and Manmupo. And of course, I wouldn't be talking about this if we couldn't, right? So we have done open modification search, right? If we don't know what modification we're looking for, we can do an open modification search. What is shown here is basically just the mass differences across the different peptides. For the species of Kopelev acidocardiaris, we actually know the great composition. So that was kind of our positive control. And then Kopelev islamicus, the related species, we didn't know the glycan composition yet, but we saw this mass difference in the high mass range. And that difference between this peak and that peak corresponds nicely to hexos. So our assumption is that Kopelev islamicus has a very similar glycan, but with an additional hexos added to it. So we'll take something like that maybe. We'll see. And then we also followed up on those results to now use this new glycan composition that we have identified from the open modification search to go back to a closed search using that glycan composition. And we do see a lot of glycans or glycosides, glycoproteins identified through this approach. So we can basically have a two-step approach where we first look with an open modification search, which type of glycan compositions we can find, and then go back into the closed search to really have a higher coverage overall of the glycoproteome. And yeah, overall, we identify a lot with the Archaeal Proteome Project. We're interested in looking at other species as well. These are the open modification search results. For most of the species, we see mass difference in a high mass range that could correspond to glycosylation. But for a lot of them, there's no similar species that we could base the glycan composition on. So we're trying to figure out how we can actually get to that glycan composition. But still promising results in that sense. Just real quick, glycoproteins are important for various different functions in the cell. Archaeans and films are glycosylated, and as mentioned before, important for the attachment of cells to hosts or abiotic cell species. AGLB is the glycosyltransferase related to SCT3 and eukaryotes, which is not really glycosylated as well. The glycoprotein is basically the cell wall of archaea. Then we have some cytochrome E. But there's a lot of glycoproteins that are proteins of unknown function, which is always a bit of a problem working in prokaryotes, especially archaea. Nobody knows what they're doing. Geoterm annotation, not really a thing. So we were also trying to figure out how we can get an idea about the function of those proteins. And that was a project of another great mathematician, Dia, who was looking at combining both sequence and structure-based tools to predict protein functions. So on the sequence level, she used ECMAP mapper to get basically the conserved domains and any core categories to get ideas about the function of those proteins. And on the structure side, now that we had output tool that is able to predict pretty well what protein structures look like, tools have come out to use these structures to compare against all the other ones. FoldSeq is one that actually works very fast and reliably in that sense. And so we can use that as well to map the predicted structures for the proteins of unknown function to maybe something that is known. So combining those two approaches, the sequence-based and structure-based approach, you can then come up with a consensus of potential protein functions. And with that, for the Archaeal glycoproteomes, we're able to find some proteases, likely proteases or peptidases, as well as some carbohydrate binding proteins. So we get at least a little bit more of an insight for what those protein functions might be, even though there's still a lot of unknowns. Less. Still a lot, but less. So making progress. And then finally, also one approach that we got into more recently is also try to compare the different data sets quantitatively. And the idea there is that we can compare the protein quantities across all of the different data sets, across all of the different conditions. We can get, again, more insights into the functions of those proteins if they're involved in some of those processes. So again, we extract the data from the product archive, go through our bioinformatic pipelines, quantify them. Challenge here is that there's a lot of different quantification methods. So we have some methods for label pre-quantification, as mentioned by a lot of students as well. We're still struggling a bit with some of the labeling approaches, but yeah, we'll hopefully get there. And then, of course, nowadays a lot of DIA that is being used that creates additional challenges on the bioinformatics side, but also really exciting results. So yeah, we'll get into those soon as well. A statistician, Kirsten, was working on this project using MS Stats to basically normalize the data sets in a way that is consistent between the different data sets. MS Stats can be used for labeled and non-labeled samples. Yeah. And kind of as a quality control, we wanted to see for proteins that show statistically significant, but the log fold changes are in a similar range. For different data sets, are we looking at vastly different fold changes, or are they all very similar? And what we see is that with this pipeline, we do get similar shapes of this histogram, similar fold changes, so that we are actually fairly confident that we could compare across those data sets. Not directly, right? We can't do a direct fold change comparison between condition 1 in one data set and condition 2 in another data set, but at least within a data set, we can do that fold change analysis and then compare the fold changes across the. This is an example of two data sets that we are comparing, again, for anaphylaxis leukemia, KC021872 and 40781. As you can see, they are not very correlated in that sense, right? We have a bunch of proteins that are important in this data set, but this line here, but not important for the 21827 data set. And that makes sense because the one data set looks at ribosome biogenesis, whereas the other one looks at cell shape differences. So again, we still need a good positive control as well, because, the data sets were generated for different biological questions, right? So it's hard to get a good positive control where it's more correlated, but at least we see that what we expected, not a good correlation, is actually what we see with the statistical analysis. Right? So in summary, I know I rushed a bit through this, but just to give a little bit of a primer basically for what we are doing in the lab as well, a lot of bioinformatics, combining the data sets. What I didn't go into is that we can also use this for proteogenomics, so improving genome annotations, especially in for prokaryotes, identifying post-translation modifications, predicting protein functions, and trying to validate those with quantitative proteomics. Right? And with that, thanks to everyone in the lab who has been working on that. And again, a lot of collaborations, a lot of other people that are involved in this, especially on the Archaeal Proteome Project side, all of the labs that are contributing the data sets to public databases. Public data is awesome. Keep doing it. It's really
yeah.
So yeah. And of course, all the open source tools as well. That's